So, it's a really exciting time to be involved in TIL therapy. This is something that I've been involved in for 10 years that I've been here at MD Anderson. And it used to be, limited to small trials, just one center at a time. And things have really evolved now with the FDA approval of a commercial TIL product. With that interest comes interest from other TIL manufacturing companies, and we have a current trial with a company called Obsidian using a novel cellular therapy product called OBX-115. What's really interesting about OBX-115 is it's a TIL cell that has been engineered with basically an on-off switch. So there is some genetic engineering of the TIL to have what's called a carbonic anhydrase drug responsive domain. So if you administer the ligand for carbonic anhydrase, which is FDA-approved drug called acetazolamide, if you administer acetazolamide after the TIL is infused, the TIL is basically turned on, and it produces its own cytokine called membrane-bound IL-15. And so the idea here is that after a patient gets their lymphodepletion and their TIL infusion, you can simply administer acetazolamide to turn the TIL cell on. It produces its own IL-15, and in that way, you don't have to give an exogenous cytokine like high-dose IL-2. I think this approach is safer because we do know that high-dose IL-2 is associated with toxicities that can limit the pool of eligible patients. And so being able to be free of IL-2 could potentially open up this therapy to a broader patient population. In this trial, I presented data on 10 patients that had been treated as of the data cut in April of this year. And we saw objective response rate of 44%. We saw no primary progressive patients, and 75% of patients were progression-free at 24 weeks. Additionally, we saw a fairly favorable safety profile. We did see some low-level toxicity, so skin toxicity, rash, itching, as well as uveitis and iritis as a consequence of the cellular therapy infusion. But otherwise, no dose-limiting toxicities. These patients were able to be treated on our hospital unit without having to go to the ICU setting. And in general, we're pretty excited about the initial results. So our current first-in-human OBX-115 trial is closed. We completed enrollment, and there is a current study, a phase 1, phase 2 study exploring OBX dosing, safety, tolerability, and efficacy both in melanoma and non-small cell lung cancer. This is a multi-center study being currently enrolling across various sites in the US. We're definitely seeing a lot more interest in TIL, not only in melanoma, but expanding to other solid tumors. So we've definitely seen some preliminary data in non-small cell lung cancer. We've seen some data in cervical cancer. And now that there is an FDA-approved product, I think we're going to see more and more interest in solid tumors beyond melanoma. I definitely think that this is an exciting time to be in TIL research, as we're seeing more and more engineered TIL that can potentially make the TIL better and hopefully also make the TIL safer. Another thing that's hugely important in TIL is trying to be able to reduce toxicity, and a lot of the toxicity that we see comes from the lymphodepleting chemotherapy. I think as we have better cells, more potent TIL, we may be able to lower the dose of lymphodepletion, which can, again, make this therapy safer, more widely available, and potentially even an outpatient therapy at some point in the future.